Assalamu alaikum wa rahmatullahi wa barakatuh. Our topic today is endocrine pancreas. Glucose is a 6-carbon molecule. It is an efficient fuel that, when metabolized in the presence of oxygen, breaks down to form carbon dioxide and water. Although many tissues and organ systems are able to use other forms of fuel, such as fatty acids and ketones, the brain and nervous system rely almost exclusively on glucose as a fuel source. So, the exclusive fuel for the brain and nervous system is glucose only. Because the brain can neither synthesize nor store more than a few minutes supply of glucose, normal cerebral functions requires a continuous supply from the circulation. It means a continuous supply of glucose from the circulation. Glucose is ingested in the diet and transported from the gastrointestinal tract through the portal veins to the liver before it gains access to the circulatory system. An important role of the liver to regulate blood glucose is through processes which are glycogen synthesis, which is called a glycogenesis. The second role is glycogen breakdown, which is called glycogenolysis, and the synthesis of glucose from non-carbohydrate sources, which is called gluconeogenesis. What that mean? When blood glucose level raised, it is removed. It means the glucose removed from the blood and converted into glycogen, which is the main short-term storage form of glucose, where this is formed. It is in the liver by glycogen synthesis, glycogenesis. This figure demonstrates glucose metabolism. When glucose is ingested in the diet, it is transferred from the gastrointestinal tract into the circulation. When there is high blood level of glucose, the glucose will be converted into a storage form called glycogen. This is done by the liver. When it is required again, the glycogen is breakdown into glucose and distributed for utilization in the circulation as free glucose. Why we don't use fatty acid as a normal source of energy in the circulation? Because fatty acids cannot be converted into glucose, the body must break down proteins and use the amino acids as a major substrates for gluconeogenesis, which means formation of glucose from a source other than a glycogen. So, the body must break down proteins and produce amino acids from these proteins as a major substrate for gluconeogenesis during period when metabolic need exceed the food intake. An example, fasting. Glucolysis is the versus of gluconeogenesis. As you see in this figure, that we need a source of protein when the intake of food is less than our needs. From where we bring this protein? We bring it from the major source of protein in our body, which are the muscles. And a protein will be break down into amino acids. These amino acids will be the basic unit for gluconeogenesis. Pyruvate will be converted into the lactate, and lactate will pass through the circulation into the liver, where lactate will convert it again by gluconeogenesis into the pyruvate, and by a process of gluconeogenesis we will produce glucose again, which will pass into the circulation and pass into the cells that need glucose. As we talking about the liver use only small amount of fatty acids from its own energy needs. I mean the liver. It converts the rest into the ketones. These fatty acids will be converted into substances called ketones. These ketones will be released into the blood circulation. In situations that favor fat breakdown, such as diabetes mellitus and fasting, large amount of ketones are released into the blood stream. Because ketones are organic acids, they will cause ketoacidosis when they are present in excessive amounts in the circulation. This figure demonstrates how our body uses fatty acids as a source of energy when there is no adequate amount of glucose present in the circulation, as we are fasting for a long period, or when there is something wrong in our body, as occur in the diabetes mellitus. When glucose isn't available, fatty acids are released from the fat cells. They are travel through the circulation into the liver, and the liver will use them to create ketones. These ketones will be used as a source of a fuel or energy for our bodies. Now, we have to understand, after this brief introduction, we have to understand how pancreas acts to produce insulin and regulate our blood glucose level. The hormonal control of blood glucose resides largely with the endocrine pancreas. The pancreas is made up of two major tissue types, the acini and the islets of Langerhans. The acini secrete digestive juices into the duodenum, and the islets of Langerhans secrete glucose regulating hormones into the blood. A glucose regulating hormones, not hormone. Each islet is composed of beta cells that secrete insulin and alpha cells that secrete glucagon, and delta cells that secrete somatostatin. Blood flows through the islets of Langerhans from the center towards the periphery. Insulin is secreted by the cells at the core of the islets, so that the alpha cells that are located at the periphery receive a high concentration of insulin. Why? Because the insulin is secreted by the cells at the core of the islets of Langerhans. This anatomic arrangement is significant because insulin and glucagon are antagonistic hormones. What do we mean by antagonistic hormones? That means that the action of insulin is the reverse of the action of glucagon. In the regulation of blood glucose. So, when the insulin is secreted by the cells at the core of the islets, the insulin will pass towards the periphery of the islets of the Langerhans. It will pass towards the alpha cells, which produce glucagon, which is the antagonistic hormone of insulin. When this high concentration of insulin pass towards these cells will be stimulated to produce glucagon. Why? To suppress insulin production in a high concentration. This anatomical arrangement is important for regulation of blood glucose and suppressing insulin from increasing in concentration. What is the function of insulin? Insulin will lower the blood glucose concentration by what? By facilitating the movement of glucose into the body tissues. It will lower the blood glucose by facilitating the movement of glucose into the blood tissue. So, blood glucose will be low, while glucagon maintains blood glucose by increasing the release of glucose from the liver into the blood. It is the reverse of insulin action. Glucagon will maintain blood glucose, not a high blood glucose, will maintain a normal blood glucose by increasing the release of glucose from the liver into the blood. Somatostatin, which is the third hormone released by delta cells, inhibits release of insulin and glucagon, and also it decreases gastrointestinal activity after ingestion of food. Why? By decreasing gastrointestinal activity, somatostatin is thought to extend the time during which food is absorbed into the blood. And by inhibiting insulin and glucagon, it is thought that somatostatin will extend the use of absorbed nutrients by the tissue. Between two practices, it will give the tissue time to absorb nutrients. Returning to insulin, insulin is the only hormone known to have a direct effect on blood glucose level. Insulin is the only hormone that known to have a direct effect on lowering blood glucose level. The action of insulin, which achieve decreasing blood glucose level, is because, first, it promotes glucose uptake by the target cells and provides for glucose storage as a glycogen. So it acts in two directions. First, it promotes uptake of the glucose by the target cells, and the second is by promoting glucose storage as glycogen. Second, It prevents fat and glycogen breakdown and inhibits gluconeogenesis. How? When insulin is present, it will facilitate the movement of glucose and the uptake of glucose by the target cell. So there is no need for fat and glycogen breakdown to produce a glucose. By this way, insulin will prevent fat and glycogen breakdown and inhibits gluconeogenesis. Finally, insulin increases protein synthesis, so it is important for muscle because it will increase the protein synthesis. Another important functions for insulin acts to promote fat storage by increasing the transport of glucose into the fat cells. And as you know, when glucose is present in a high concentration, it will be converted into the fat in the fat cells. Also, glucose facilitates triglyceride synthesis from glucose in the fat cells and inhibit the intracellular breakdown of stored triglyceride. Insulin also inhibits protein breakdown because it increases protein synthesis by increasing the active transport of amino acid into the body cells. Also, insulin inhibits gluconeogenesis, or the building of glucose from new sources, mainly amino acids. When it is present in a sufficient amount, when glucose is present in a sufficient amount and insulin present in a sufficient amount, protein breakdown is minimal. Why? Because the body is able to use glucose and fatty acids as a fuel source. In children and adolescents, insulin is needed for normal growth and development. This figure demonstrates how we use insulin to produce proteins, store glucose in the liver as glycogen, and also how insulin help the body in producing triglycerides, and how glucose pass into the fatty cells to be stored as fatty acids. Now, we have to know how insulin is synthesized and released into the circulation. As we remember that insulin is produced from beta cells in the islets of Langerhans. The active form of insulin is composed of two polypeptide chains, the A chain and the B chain. Active insulin is formed from a large molecule called the proinsulin. In converting proinsulin into insulin, enzymes in the beta cells cleave proinsulin at a specific site to form two separate substances, which are the active insulin and a biologically inactive connecting peptide, which is called C-peptide. The inactive form of a hormone is composed of two polypeptide chain, the A and beta B chain. It is produced from a large molecule called proinsulin. When the body needs insulin, there will be conversion of proinsulin into insulin. By what? By enzymes in the beta cells, which are cleave proinsulin at a specific site to form two separate substances, the active form of insulin and the inactive connecting peptide, which is called the C-peptide chain, that joined the A and B chain before they were separated. This figure demonstrates the insulin molecule, which are composed of two polypeptide chains. This is the proinsulin and this is the connecting peptide. Why it is called the connecting peptide? Because it connects the two polypeptide chains. This is the A chain and the B chain. When the connecting peptide is present, the molecule is called a proinsulin. When the body needs insulin, there will be disconnection between the connecting peptide and the proinsulin. There will be a two molecule. The first, which is the connecting peptide, which is inactive, and the other is the active insulin. Okay, till now. Active insulin and the inactive C-peptide chain are packaged into a secretory granules. They will not released immediately. They will be packed into a secretory granules and released simultaneously from the beta cells. What do we mean by simultaneously? They will be released as the active insulin and the inactive C-peptide in the same time from the secretory granules. The C-peptide chain can be measured clinically. Why? This measurement can be used to study beta cell activity. For example, injecting exogenous insulin in a person with type 2 diabetes will provide few or no C-peptide chain. Why? Because it is exogenous. It is exogenous from the outside of the body. When we inject insulin into a person with type 2 diabetes, this injection contains the active insulin only. So, if we do measurement for the insulin and C-peptide, there will be a few or no C-peptide chain, whereas insulin, which is endogenous, secreted by the beta cells, would be accompanied by secretion of C-peptide chain. What do this mean? This means that when we need to measure the beta cell activity and the presence of insulin, the endogenous insulin that our body produces, we will measure the C-peptide chain. If it is present in a normal concentration, that means the beta cells are actively producing insulin and secreting it into the circulation. The release of insulin from the pancreatic beta cell is regulated by blood glucose level. It is regulated by blood glucose level. Increasing blood glucose level will raise and decrease when the blood glucose level decline. When there is a high concentration of blood glucose, the level of insulin will be increased. While when there is low concentration of blood glucose, the level of insulin will be decline. Secretion of insulin occurs in an oscillatory or pulsatile fashion. What do this mean? After exposure to a glucose, which is called the first phase release, of stored preformed insulin. Preformed, that means that the insulin is formed in the beta cells and stored in the secretory granules, followed by a second phase release of insulin, which is a newly synthesized insulin. A serum insulin level begins to rise within minutes after meal, reach to a peak in approximately 3 to 5 minutes, and then it will return to the baseline level within 2 to 3 hours. What do this mean? This means that when we start eating and complete our meal, the insulin level will increase within minutes and reach to its peak level at 3 to 5 minutes. And then after 2 to 3 hours, the insulin level will return to its baseline level. Insulin secreted by beta cells enter to the portal circulation and travel directly to the liver, where approximately 50% is used or degraded. Insulin, which is rapidly bound to a peripheral tissue or destroyed by the liver or the kidney, has a half-life of approximately 15 minutes only once it's released into the general circulation. The half-life of insulin is 15 minutes only. It will not remain in the circulation for a long period. This figure demonstrates the beginning of insulin release and the phase 1, in which insulin reached to its peak level within 3 to 5 minutes. This is called the phase 1 release. Another figure this is the baseline concentration in the circulation. Insulin will be released after 3 to 5 minutes from completing our meal, which is called the first phase or acute phase release. Okay, and the second phase release after 2 to 3 hours. By the way, insulin is circulated unbound in the circulation, and it will be cleared from the circulation within 10 to 5 minutes. How insulin acts on the target cells? And is insulin having a specific receptors in these target cells? To initiate its action on the target cell, insulin will bind to activate a membrane receptor. So receptors for insulin are present on the cell membrane. It is the activated receptor that is responsible for the cellular effect of insulin. What is the composition of insulin receptor? Insulin receptor is a combination of 4 subunits. A large alpha subunit that extend outside the cell membrane, and it is involved in the insulin binding. A large alpha subunit extend outside the cell membrane and is responsible for the insulin binding, while a smaller beta subunit that is predominantly present inside the cell membrane and contains a kinase enzyme that becomes activated by insulin binding to the large alpha subunits. Activation of kinase enzyme results in phosphorylation of beta subunit, which in turn activates sub-enzymes and inactivate other. The activation of kinase enzyme will result in phosphorylation of beta subunit, which in turn activates sub-enzymes and inactivate other enzymes, thereby directing the desired intracellular effect of insulin on glucose, fat, and protein metabolism. As you see, this is the alpha subunit, which is protruded outside the cell membrane, and it is involved in the binding of insulin, while the beta subunit present embedded inside the cell membrane, and it is responsible for the activation of the kinase enzyme and the corresponding phosphorylation inside the cell. Another figure which demonstrates how insulin will act on its receptor. As you see, this is an important figure. The alpha subunit, which are present outside the cell membrane. This is the molecule of insulin, which connected to the alpha subunit. When there is a connection of insulin to its receptor site, there will be activation of beta subunit, which contain the kinase enzyme. When activation of beta subunits occur, there will be activation of kinase enzyme, and there will be activation of another enzymes inside the cell. And as you see, this connection of insulin hormone to its receptor will allow opening of cell channels inside the cell membrane, and glucose will pass directly into the cell. As you see, glucose will not pass through this insulin receptor site. Glucose will pass through a specific channels from outside the cell into the inside of the cell by a specific glucose transporter. We will learn about these transporter next in the lecture. Another channels for passing of amino acids, as we said earlier in the lecture, that that insulin allow production of proteins by allowing movement of amino acids into the target cells. So, after activation of insulin receptors, kinase enzyme will activate a series of phosphorylation, and the resultant is passing of glucose into inside of the cell and passing of amino acid into inside of the cell. This will cause enzymatic activation and deactivation that results in glucose transport into inside of the cell, glycogen synthesis, and you know where glycogen is synthesized and stored, fat synthesis when the glucose passes into the fatty cells, protein synthesis when the amino acids is passed into the muscle cells, and also growth and gene expression for a new protein synthesis according to the need of the target cells. Now, as you remember in the last figure, we talk about glucose transporters. So, we have to understand what are these glucose transporters. Because all membranes are impermeable to glucose, they require a special carrier called glucose transporter to move glucose from the blood into the target cells. Within seconds after insulin binds to its membrane receptor, the membranes of about 80% of body tissue increase their uptake of glucose by means of special glucose transporters. This is particularly true for skeletal muscles, adipose tissue. Considerable research has revealed a family of glucose transporters termed as glucose transporters 1, 2, and so forth. For example, a glucose transporter 4 is the insulin-dependent glucose transporter for skeletal muscles and adipose tissue. It is sequestrated inside the membrane of these cells, I mean the skeletal and adipose tissue, and thus is unable to function as a glucose transporter until a signal from insulin causes it to move. Signal from insulin, it means binding of insulin to its receptor site. This will cause it to move from its inactive site into the cell membrane, where it facilitates glucose entry into inside of the cell. Glucose transporter 2 is another example of glucose transporters. Is the major transporter for glucose into the beta cells and liver cells. It has a low affinity for glucose and acts as a transporter only when plasma glucose levels are relatively high, such as after meal. What do this mean? This means that this transporter will not act in the normal condition. It will act when the blood glucose level is very high and the target cells took their requirement while blood glucose level remain high. So in this condition, the glucose transporter 2 that are present in the beta cells and liver cell will act to transport glucose from the circulation into the liver cell so that the glucose will be converted into glycogen, and also it will pass glucose into the beta cells. Why? You will answer this question. Another example of glucose transporters is glucose transporter 1, which are present in all tissue. All tissues in our body have a glucose transporter 1. It does not require the action of insulin, and it is important in the transport of glucose into the nervous system. It is important to transport glucose into the nervous system. That is why it is not require the action of insulin. And examples of the glucose transporters in our body and their sites. As you see, glucose transporter 1 present in the blood-brain barrier, in the heart and in the blood. The type 2 glucose transporters are present in the liver, pancreas, and small intestine. The type 3 in the brain and nervous tissue. The type 4 in the skeletal muscle, adipose tissue, and the heart, and etc. Now, we have to understand how insulin acts in the normal physiological condition and why it is unable to act in diabetes mellitus type 1 and type 2. In the normal physiological condition, insulin binds to insulin receptors. Insulin will bind to insulin receptors and triggers the opening of glucose transporters in the fat and muscle cells, allowing glucose removal from the bloodstream, and so glucose will pass into inside of these cells. While in type 1 diabetes, insulin is not produced. So when there is a high concentration of glucose in the circulation, insulin is not produced from the pancreas. So the insulin will not activate its receptors. In type 1 diabetes, there is no insulin to activate insulin receptors, so that the glucose will not removed from the bloodstream and remain in a high concentration in the bloodstream. Why? Because insulin is not present, so insulin will not activate its receptor, and there is no consequent activation of glucose transporters. So glucose will remain in a high concentration in the circulation, causing diabetes. While in type 2 diabetes, as you see, there is a high concentration of insulin, but prolonged overproduction of insulin. In type 2 diabetes, there is a prolonged overproduction of insulin, lead to desensitization of insulin receptors, and hence glucose is not removed from the bloodstream, causing diabetes. Insulin is present in a high concentration, result in desensitization of the insulin receptors. The insulin receptors are not sensitive to insulin, so there is no activation of insulin receptors, and subsequently there is no activation of glucose transporters, so the glucose will remain in the circulation in a high concentration, although there is a high concentration of insulin. This condition is called insulin resistance. Another figure that explains the pathogenesis of type 2 diabetes mellitus. Now, the antagonist hormone of insulin is glucagon. It is important to have some information about this important hormone. Glucagon is a polypeptide molecule produced by the alpha cells of islet of Langerhans. It maintains blood glucose between meals and during period of fasting. It is important for our bodies during fasting and the period between two meals. Why? To maintain normal blood glucose level. Like insulin, glucagon travels through the portal vein into the liver, where it exerts its main action. So its action will mainly occur in the liver. Unlike insulin, glucagon produces as increase in blood glucose. As you remember, insulin is produced when there is a high blood glucose level, so insulin will lower the blood glucose level, while glucagon produces an increase in blood glucose level. Antagonistic action. The most dramatic effect of glucagon is its ability to initiate glucogenolysis, or breakdown of the glycogen in the liver. The most important action of glucagon in maintaining normal blood glucose between meals and during period of fasting is by increasing blood glucose level. By what? By glucogenolysis. How? It will act on the liver cells to break down the glycogen in the liver as a means of raising blood glucose. Okay? Usually within a matter of minutes. Because liver glycogen stores are limited, so the liver will need gluconeogenesis. Why? Because glycogen stored in the liver is limited. So glycogenosis is an important role in maintaining blood glucose level over time. Glycogen also increases the transport of amino acids into the liver, and glucagon also increases the transport of amino acids into the liver and stimulate conversion of amino acids into glucose. As with insulin, glucagon synthesis and secretion is regulated by blood glucose. The two hormone concentration is regulated by blood glucose level. Decrease in blood glucose concentration to a hypoglycemic level, not to a normal level, to a hypoglycemic level, produce immediate increase in glucagon secretion, and an increase in blood glucose to a hyperglycemic level produce a decrease in a glucagon secretion. As we said, the glucagon secretion is regulated by blood glucose. So a hypoglycemic level will immediately increase glucagon secretion, while a hyperglycemic level will produce a decrease in glucagon secretion. High concentrations of amino acids as occur after protein meal also can stimulate glucagon secretion. In this way, glucagon increases the conversion of amino acids into glucose as a means of maintaining the body glucose level. Glucagon levels also increase during strenuous exercise as a means of preventing a decrease in blood glucose. These are some figures that illustrate the action of insulin and the glucagon. Finally, thank you for your attention and goodbye.